This is actually not as advertised. I was going to talk today about clinical genetic databases, which I think are a sort of really interesting new type of biobank that we can use for research. But after sitting in this room for the last several days and hearing about people's phenotyping dilemmas and the various research projects, I just thought it would be much more useful for everybody, your time, for me to talk about Phecodes for hopefully about a little over 20 minutes and just give the basics, because I think for anybody who's doing biobank research, I really do think that a familiarity, at least a passing familiarity with Phecodes and use of Phecodes can help solve some very basic problems that you run into and help increase the rigor of your research. So before I get into the definition of what Phecodes are, I want to point out why I think this is relevant for this room. Phecodes, they're phenotypes that are compatible with essentially all biobanks. Any biobank that has EHR records linked in, you can use Phecodes on. And so this is a phenotyping tool that just has a lot of relevance and can really help with cross biobank studies. So what are Phecodes? Wait, how many of you have run a PheWAS? Yeah? How many of you have used Phecodes? Okay, if you've run a PheWAS, you've used Phecodes, probably. Although the definition of what a Phecode is or what a PheWAS is, sometimes I'm asked to be a reviewer on papers just to adjudicate whether what they did was a PheWAS, like if it counts. PheWAS stands for a phenome-wide association study. The idea of a PheWAS is that it's a kind of an analog to GWAS, where you take an exposure, typically a genetic exposure, and scan across a medical phenome and look for associations. And in order to do that, you need a way of basically ascertaining case and control status across a really wide swath of phenotypes. And that's what Phecodes do. So the way that they do this is that they use ICD codes as the basis of case control definition. As you probably know, there are tens of thousands of ICD-9 codes and 10 codes. Specifically, there are about 15,000 ICD-9-CM, 90,000 ICD-10-CM. You could, of course, test those individually, but that would create a really large multiple testing burden. And there are a lot of ICD codes that are basically pointing to a very similar thing, which I'll show you in the next slide. And so what we did with these to create Phecodes was, when we created these, actually it was back when there was only ICD-9. We wanted to condense all of these ICD-9 codes, condense some of the details, and group them together so that we had clinically meaningful phenotypes. So the version of Phecodes that most people use, version 1.2, has a little over 1,800 Phecodes. And by the way, Jana, I noticed too, you didn't have any ICD-9s in your CAD definition, so I was curious about that.
James told me this week that there are new phenotyping going on in all of our including ICD-9.
Interesting, yeah. So most biobanks, all of us included, not UK Biobank, which is a little bit different. They only use ICD, the ICD-10, sort of WHO version of ICD-10. But most US biobanks, if they have any longitudinality, will have both ICD-9 and ICD-10. That can be a real pain in the tail, even if you're just doing an individual phenotype, because you're going to have to deal with two different coding systems. But Phecodes, we do our best to kind of reconcile those two and map between those two so that you can just have one unified system of phenotypes. So here's just a really quick example, a great case in point of the kind of condensing that that Phecodes does. This is an example from ICD-9s. I don't know if you guys have ever read ICD-9s starting from the beginning. I think 001 is cholera. These ICD-9s have a very interesting historic legacy. And so a lot of the early codes are actually pertaining to infectious diseases that were really of public health interest back in the day, like 100 years ago. And reflecting the sort of interest in what people were trying to catalog many, many years ago, there are actually 496 different codes that essentially are different forms of tuberculosis. None of this granularity is particularly relevant unless you're doing a very, very specific study. And so all of these codes are actually just mapped to a single Phecode for tuberculosis. And again, mapping in ICD-10s as well. This is a chart that I produced for a review paper, which I should have cited here. I wrote a paper for annual reviews a few years ago where I just wrote down everything that I knew and learned about Phecodes. And so that's a nice reference material, which you can get on our website, which I'll show you in a minute. But here just shows that Phecodes are broken up into different chapters. These chapters are not even loosely based. I think they're precisely based on ICD-9 chapters. So you have a chapter for infectious disease, endocrine metabolic, neoplasms, etc. And you can see here that the number of Phecodes is variable across these chapters. There's in the Phecode 1.2 version, there's not that many infectious disease codes compared to the endocrine metabolic. This is a really important slide here. So when you use Phecodes on your data set, you take all these ICD codes and you can, label them as yes, no, you have any one of these set of, let's say, ICD codes for asthma, right? But what we found, and this result tends to be pretty robust, is that requiring a Phecode on at least 2 unique dates vastly improves the performance of the phenotype. This is because in certain contexts, particularly in the inpatient context, ICD codes can be applied to people's medical record for disorders that are suspected but haven't been clinically confirmed. And so there are situations in which somebody may have an ICD code for, let's say, CAD, but that code is being applied, let's say, because they came into the emergency room with chest pain and they were going to get some sort of workup to determine whether they had CAD or maybe troponin for MI. So these multiple days can clean up Phecodes, or the positive predictive value specifically of these phenotypes. So this chart here, this is from a really old poster actually that we presented at AMIA way back in the day, where we looked at a number of different conditions: atrial fibrillation, Crohn's disease, multiple sclerosis, etc. And this is just the F1, the harmonic mean of the PPV and an NPV. And what we found for these diseases, these 4 diseases that we looked at, was that over all of these, if you average across all these diseases, the maximum F1 score was at 2 or more. This is in a lot of papers now. I think my colleague Scott Hebring named this. They call it the rule of 2. We use the rule of 2, meaning 2 or more unique dates qualifies for a case. Individuals with 1 Phecode, only 1 date, are oftentimes excluded from the analysis, and then everybody else with 0 codes is a control.
What would you consider a different date, like a week? A different
I literally do unique by date.
Okay.
But of course you can play around with temporal measures, but that's the end of my talk, I think, will be informative because, yeah, I guess I will take that as an invitation to make a really important point. The performance of these Phecodes, the ideal minimum code count, is going to depend on a lot of factors, right? One, you think maybe acute conditions, maybe you're going to lose a lot of cases if you require 2 or more codes, let's say for strep throat or something like that. For chronic conditions, you might be able to require 4 codes and not you lose that many true positives. You see what I mean? The longitudinality of your cohort is going to probably change the optimal minimum code count. So if you have 30 years of records and you're looking for a chronic condition like multiple sclerosis, you might be able to require 10 codes and not lose that many true positives. You see what I'm saying? So it is in some ways cohort dependent, although this rule of 2 thing is one of these things that has cropped up again and again in many different cohorts and across many phenotypes. Go ahead, Haki.
So why does it come down when you have more?
Because when you have more, and this is particularly true for atrial fibrillation, which is more of typically is an acute condition, the F1 score is a harmonic mean between the positive predictive value and the negative predictive value. So basically, there are a bunch of people when you require 3 codes that actually have atrial fibrillation, and you're classifying as negative, basically. Well, if you actually used it, you would exclude it from the study.
Here is you are requiring this.
Yeah. You're losing true positives. You're losing true positives. So yeah, and so, if your goal is to balance the NPV and the PPV, and we see this again and again, and this plot doesn't make it as much dramatic because of how far down this AFib thing goes, but we typically see a really good jump of PPV when we go from 1 code to 2 code, and then a more shallow increase in positive predictive value.
At an individual level, the more codes instances you see, the more likely the person does have it.
Yeah, exactly. It's increasing the probability that they actually are affected. Okay, so back in the day when we started doing a lot of this work with PheWAS, and this continues on today, there were a lot of concerns and maybe even kind of aggravated complaints about the idea of basing research on ICD codes, because the fact of the matter is that these ICD codes are not applied to medical records or attached to medical records for the purpose of research. They're used for reimbursement, right? So there's a whole financial incentive that is wrapped around the use of these codes, which clearly is going to affect the way that they're used and applied. And so I completely am sympathetic to the skepticism of using these types of ICD codes. But this paper in 2013 that we put out really made me a believer that there was a lot of meaningful information that's in these ICD codes, which again, you can condense to Phecodes. And what we did in this paper is we took all of the associations that were in the GWAS catalog at the time. Many of these were from pretty high quality studies where they actually interviewed the patient or phenotyped the patient personally. There was some Wellcome Trust data in there. So this was 2013 GWAS catalog. Every disease that was in the GWAS catalog at the time that we were able to represent as a phe code using ICDs, we attempted to replicate that association. And what we found was that among the associations that we were powered to replicate, we replicated about 3/4 of them, which was far, far beyond chance. And not only that, but you can see here, and we did use the rule of 2 for those. And this was across multiple sites, 5 different sites, by the way. This wasn't just Vanderbilt data. You can also notice that our replication here, this is log P at the bottom, these are all of our replication attempts, is that we were able to replicate things from cancer to endocrine problems to dermatological problems, et cetera. It wasn't just one body system. So this really shows that Phecodes do have this ability to kind of robustly replicate all of these association studies, which indicates that they do have really valuable information content, despite the fact that you have to acknowledge that there's some noise. Okay, so what are some of the fun things that you can do with Phecodes? First of all, very quick, oh wait, maybe I'll look at these are the fun things. Okay, I'm gonna go through them. Number 1, PheWAS. So for the non-initiated, the idea of a PheWAS is that you can take an individual, in this case, this is a genetic variant, a common genetic variant that we found associated with hypothyroidism. A big GWAS we did across 5 medical centers for hypothyroidism. And then what we did was we took this one SNP and we ran a PheWAS. So we looked for associations across the whole medical phenome, right? And saw if there was any other pleiotropic associations that we could identify with this SNP. This PheWAS isn't particularly interesting, although you can see here, if you can read thyroiditis and non-toxic nodular goiter, these phenotypes did also show up as significantly associated with this variant, and we replicated the hypothyroidism association there. This is a better looking, so initially when people started PheWASing, when we put this method out, it was a very popular way of looking at common genetic variants in pleiotropy. But it didn't take too long, and this is something that came out of Mount Sinai, Emer Kenny and others, who started looking at rare genetic variants and doing PheWASs, so very similar principle, but instead of having a common SNP, she looked at a functional variant. This is a really wonderful study where they found a variant in a collagen gene that I think was very common in Puerto Rico, if I recall correctly, that had a number of really robust phenotypic correlations that they were able to find with this PheWAS. You can download our we've been trying to put out a legit R package in CRAN for like a long time, and it just keeps getting kind of backburnered. But there is a PheWAS package in R that you can use to run PheWAS. There's also a Python version that folks at the NIH have developed. And you could always talk to me if you're interested in running PheWAS. It's very easy. It's really just a for loop. There are also a lot of really great resources that have been developed around Phecodes and PheWAS. This is my absolute favorite. The University of Michigan has a website called PheWeb that you may want to look at if you have any variants that you're associating or that you're looking at. So this is for the UK Biobank. They ran a PheWAS by GWAS. So basically they imputed all these variants in the UK Biobank cohort, and then they encoded all the UK Biobank phenotypes into Phecodes version 1.2, and they did an all by all. And so this is just a wonderful interface that you're able to look up by gene, by variant, or by phenotype and get either Manhattan plots or PheWAS plots. Highly recommend that. People also do associations with non-genetic exposures with Phecodes as well, and PheWAS. Here's an example of a paper we put out a few years ago where we were looking for novel risk factors of something called bronchopulmonary dysplasia. This is a problem that happens in premature babies where their lungs are not fully developed. But there's a lot, not all babies who are born premature have BPD or bronchopulmonary dysplasia. And so there's some sort of unaccounted variance. It's not fully understood why some of these babies develop this. So in this case, we looked at prenatal diagnoses that occurred in the pregnant mother, and we looked at if there was anything that was associated with the outcome in the baby in a linked database. And we found, and we replicated this in a separate data set as well, that moms who have polyhydramnios, that means having a lot of extra amniotic fluid, they had a much higher likelihood of having a baby with BPD. So this is just one example of many of non-genetic exposures that people study with PHEWAS. And finally, this is a study where we looked at, oh, this is the wrong citation. This was in JAMA Oncology, I think. Chenji Zheng is the first author, excellent researcher at the NIH. We've also run PHEWASs where instead of looking at an individual variant, we're looking at individuals who have pathogenic variants and known hereditary cancer that are associated with hereditary cancer syndromes. So for instance, we take all the individuals with a CHEK2 variant that's known pathogenic, or these Lynch syndrome genes, et cetera, and ran a PHEWAS. And this plot here is showing all of the gene groupings that we did. And the blue ones here, or sorry, the black associations here are very well-established kind of replications of the different phenotypes that are associated with these cancer syndromes. The reason we ran this study, by the way, which I think is a little bit of a side note, but I think is like kind of an interesting rationale for the study, is that, you know, there's all these cancer risk genes, right? Like BRCA1 and BRCA2, I'm sure everybody's heard of, right? PALB2, APC, et cetera. Most of these syndromes are really only known for their propensity to give people a risk factor for cancer, various cancers, sometimes prostate cancer, sometimes stomach cancer, et cetera. But we wanted to test the hypothesis that there was maybe additional phenotypes that were non-neoplastic in nature that characterize these syndromes. And I think we did have some interesting findings, but I'm going to have to leave you hanging because I have a few more things to talk about. Okay, the next application where you can have fun with Phecodes is that you can start, so you can run a phenotype, or a phenotype risk score, for which we have also a nice R package. And very briefly, the idea of a phenotype risk score is that if you take a monogenic condition like Fragile X, like Huntington's, like cystic fibrosis, like sickle cell anemia, many of these disorders present with phenotypes that cross multiple organ systems. This is an example of cystic fibrosis. You can see here that it really is characterized by a bunch of different lung dysfunction, bronchiectasis, asthma, et cetera, as well as pancreatic insufficiency, male infertility, dehydration, et cetera. What we did was we created a mapping of HPO terms to Phecodes. And what that meant is that we could represent any syndrome, any genetic syndrome, in the language of Phecodes. What that allows us to do is actually look for these patterns in biobanks. So we could take the phenotypic patterns of cystic fibrosis and look in UK Biobank and say, who sort of most closely resembles this phenotypic picture? Again, we do have a legit R package in CRAN, produced by the wonderful Layla Ref, one of my PhD students, who's an excellent programmer, if you want to try that out. And this package also includes the HPO mapping, which I think in and of itself could be very useful for certain projects. One more application that I think is quite interesting and helpful with Phecodes is that because they can be used across biobanks, you can create these prevalence maps using Phecodes where you can compare the prevalence of different conditions across different biobanks. Why would you do this? Well, when I get a new data set that's an EHR data set, I create one of these prevalence maps because it just is a quick way of kind of getting an understanding of like what are the comorbidities that are occurring in this population? What's the morbidity look like globally? And because a lot of these maps have been produced, and I believe they think they're published in one of the packages that we put out, you can then say, well, what's the prevalence of hypertension in our cohort compared to UK Biobank, compared to all of us? So this is a plot here where we're looking at BioView, that's Vanderbilt's biobank, all of us, MGI, that's my friends at Michigan, the biobank that they have there, and UK Biobank. And you can see here that for some conditions, there is a vast difference in terms of the prevalence of certain conditions. For instance, MGI and BioView have a way higher prevalence of hypertension. That is a reflection of the fact that we have a much more sick population. We have a hospital-based population at MGI and Vanderbilt compared to the much healthier UK Biobank, which is actually a cohort that is healthier than the average inhabitant of the UK. This is just a really quick tutorial, just like as like nuts and bolts of like how do you actually make Phecodes. Okay, you start with a table of ICDs. These are ICD-10s here, right? So you get a column for ICDs. You have a flag, which indicates whether it's an ICD-9 or a 10. And then you have a Phecode. You can download this map file from our website, which I'll show you in a minute, or from our GitHub. And then here, you got your data. This is what you have to provide. Person ID, this is the date when that's attached to the ICD code, the ICD itself, and a flag. And then you see where I'm going here. You can do a merge. You can join it. This is a SQL code where you can just do a join, simple join. You could also do this in R using merge. I've never written it in Python. I'm not a big fan of it. I mean, Python's fine. I just typically write things in R. But there's really nothing more to it. I'm not sure if this slide is even very useful, but maybe this makes it a little bit more concrete about how you would actually create these things. Okay, and the last, oh, perfect. I have like 5 more minutes. I want to talk really quickly, Jana, I think something that maybe you were actually doing kind of intuitively in your last presentation, which is like, okay, first of all, you were like, let's just look at this GWAS for CAD and all of us. And you're like, holy crap, where are all the peaks? There's nothing there, right? This is a big red flag for you in your research. And so then you began to conduct experiments where you modified the cohort, the covariates, and the phenotype definition, and you tested the capacity to replicate certain associations. So it's that very intuitive research process that we tried to leverage in the creation of something we call the phenotype-genotype reference map, which I hope is a very useful tool in order to do replication experiments, oftentimes around questions of data quality. So the premise and part of the phenotype-genotype reference map also is based on a kind of scary fact, which is that while we have very sophisticated, well-established quality control metrics that have been developed for genotype data, there are no quality control metrics that have been standardized for phenotype data. That's a bit scary, and it's a big, big gap in the field as so much of our work in genetics is moving towards biobanks. Traditionally, in the eMERGE network, one of the things that we would do is that we would try to make a phenotype algorithm or Phecode, and we compare it against what adjudicated gold standard in the charts, right? So we'd have a clinician read that, and then they'd have a gold standard, and we compare that. That is not possible in all of us. That is not possible in UK Biobank because we do not have access to the notes. And that leaves us with a question of whether our phenotypes have any meaning whatsoever. So how do you figure that out? So there is this idea, though, of using replication as a proxy measure for data quality. So in any large biobank, the thinking goes, you should be able to replicate many of the known genotype-phenotype associations that have already been published. And if you find that you have a low replication rate or you fail to replicate for a particular phenotype, that may be, that's kind of a red flag that you may have a problem with phenotype quality. I'm not going to go through this. This is an interesting paper talking about replication back on the dawn of GWAS. I'm not going to talk about this too much, but except when we created the PGRM, it was pretty labor intensive because we went through every study in the GWAS catalog, and we really tried to curate and identify studies that were really fit for this type of replication experiments. For instance, there may be studies in the GWAS catalog that are labeled as CAD, okay, as coronary artery disease. But then when you actually look at the paper, which we, mostly me, looked at every single one of these GWAS papers, okay, to figure out like what did they actually do. Sometimes you'll find it's like people with CAD who are smoke 2 or more packs a day, okay? Like that's kind of a different cohort. That's kind of a different phenotype. It's kind of, this is a bit of a bad example. They'll be like breast cancer. That's the phenotype. This is an association with breast cancer. And then you look at the paper and it'll be like breast cancer in BRCA1 carriers. Okay, that's a different phenotype. So what we did when we matched EFO codes from the GWAS catalog to Phecodes, but we really attempted to only include studies that really represented like, you know, non-modified phenotypes ascertained on the adult population, if that makes sense. We also did a lot of allele flipping and blah, blah, blah. So this is what the PGRM looks like. Again, we have a package on GitHub that's available that you can download. We encoded all of the SNPs in HD19 and then 37 as well. We encoded the ancestry of the discovery cohort, and did allele direction, and then we mapped everything to Phecodes. So these are associations that you should be able to, I'm not going to go through this too much, that you should be able to replicate. And then in the paper that we put out a couple years ago now in AJHG, we attempted to replicate associations across 4 different cohorts 2 BioView cohorts, a European and African ancestry cohort, MGI, UK Biobank, and Biobank Japan, and we investigated replication. But in the waning 2 minutes, you know, so you were saying, look, it's really expensive. We're doing all these studies where we're trying to figure out the CAD phenotype. One way to save money is if you go into or into All of Us or UK Biobank and you have concerns about the phenotype that you're studying, like CAD, I would encourage you to just download that PGRM and make a file of the 5,000 variants that are in there. You know, that is circa 2023, all the CAD variants that have been reported in the GWAS catalog. And then run your association studies on that set of 5,000 variants, as opposed to an entire chromosome or, God forbid, the whole genome, because that's going to be very computationally expensive. One thing that we looked at, which is an important premise for this, is, you know, this is only going to work if the PGRM, if this replication as quality metric is sensitive to noise. So we took phenotypes and we injected like 10%, 20%, 30% random noise into them. So we randomly shuffled the case control status. And what we found was that our replication rate monotonically decreased as we increasingly added more noise, which means that this is a sensitive measure, and it's sensitive to at least 10% noise injection. Oh, this is a bit niche about exclude ranges. I'm not going to talk about it. But yeah, basically, one thing that I think is really neat about the phenotype genotype reference map and looking at replication as a metric is that you can start testing different assumptions. Let's say you don't want to just run your paper, I'm going to use the rule of 2. You want to say, well, is the rule of 2 actually better? And how do I actually evaluate that across so many phenotypes? One way you could do it is you could test how the minimum code count affects your capacity to replicate known associations. And you can see on this slide here that the percent of associations that we replicate, you look at that big jump from 1 to 2. It increases vastly when we require 2 or more phecodes. And you can see actually that the replication rate increases until about 5 and it kind of stabilizes. The bottom one shows kind of loss in terms of negative predictive value. Here's our GitHub page. This one's on the Huey Lab website. I won't talk about this right now. That's a bit too long. Very seriously, last thing. Okay, when we made Phecode 1.2, I initially said we wanted to group things into clinically meaningful concepts. That is a pretty, what does that even really mean? Those are just words, right? What we were really trying to do was we were trying to represent in an individual phenotype the type of stuff that people were interested in doing GWAS on. And at the time, that was common complex disease that was found in adults, Crohn's disease, rheumatoid arthritis, lupus, etc. But there is more information that is available in ICD codes than just those common complex diseases. There are a lot of congenital anomalies. There are a lot of infectious diseases. There are a lot of complications of pregnancy that didn't at the time and still don't have very good representation in the GWAS catalog. But those phenotypes are really important if you're going to be studying, let's say, perinatal outcomes, which we're interested in, or monogenic conditions. For that reason, we created a new version of Phecodes called phecodeX, which increases the granularity of phecodes and particularly attempts to represent the types of concepts that are in the human phenotype ontology, not the GWAS catalog. So I've switched over to phecodeX, the newer versions of All of Us, where they're doing all by all stuff. They're going phecodeX as well. So I think it's increasingly kind of getting uptaken by the community. And finally, this is one of our websites, our main website, where you can download phecodeX, Phecode 1.2, all the mapping files I referred to later. There are also some PWAS catalogs there, etc. And I apologize, I went 1 minute over, but I guess I could just talk about Phecodes forever. I hope you guys learned something helpful here. Just use them. I'm not getting royalties from this. They're helpful. Okay, thanks so much for your attention.